Welcome back to Module 3, where we're now going to be looking at Chapter 20 entitled Antimicrobial Medications. Specifically, we're going to look at sections 20.1 through 20.3. And these were our topics. We're going to look at this brief introduction of the discovery of antimicrobials. We'll consider some terminology, bacteriostatic versus bactericidal. We'll look at the 6 targets of antimicrobials, and then we're going to consider some safety and side effects. So let's go ahead and get started. Let's discuss a bit of history as it pertains to the development of antimicrobial agents. At the beginning of the 20th century, much of medicine was involved with diagnosing illness, describing its expected course, and telling family and loved ones just how long a patient might be sick or when they might expect a loved one to die. Even with the germ theory of disease and knowing the cause of many disease, this is what we were looking at the beginning of the 21st century. We had little that could be done to inhibit the growth of a pathogen. It was at this time that Paul Ehrlich, a German scientist, proposed chemotherapy to describe the use of chemicals that could selectively kill pathogens while having very little or no effect on a patient. His search for antimicrobial agents resulted in the discovery of an arsenic compound that worked against the bacterial agent that caused syphilis. In 1908, he was awarded the Nobel Prize for Physiology and Medicine for his discovery. A few years later, a Scottish physician and scientist, Alexander Fleming, discovered the antibacterial action of penicillin released from Penicillium, a mold which created a clear zone where bacteria couldn't grow. And this was an accidental discovery in 1928. As a physician and not a trained researcher, Fleming's initial interest in infection really stemmed from his experience in World War I, where he observed the death of many of his fellow soldiers that weren't always the result of wounds inflicted in battle, but from the ensuing infection that couldn't be controlled after. In an article he wrote during this time, he discussed the presence of anaerobic bacteria in deep wounds, which proliferated despite antiseptics. His research wasn't initially accepted, but undaunted, he continued his work and research as he still continued to practice medicine. He published his discovery of penicillin in 1929, when the scientific community greeted it with very, very little enthusiasm. And it wasn't until 1940 when 2 other scientists became interested in penicillin and were able to mass produce it that ultimately led to Fleming's winning of the Nobel Prize in Physiology and Medicine in 1945. Though arsenic compounds and penicillin were discovered first, they weren't the first antimicrobials in widespread use. Ehrlich's arsenic compounds were toxic, and penicillin wasn't available in large enough quantities to be all that useful. This brought forward another German scientist, Gerhard Dominck. He discovered a practical antimicrobial agent, the sulfonamides, in 1932. And this was the first practical antimicrobial agent used to treat a wide variety of bacterial infections. And this discovery became the basis for a number of today's sulfa drugs, the first type of antibiotic. And he won the Nobel Prize in Physiology and Medicine in 1939 for his work. So I introduced these people to you, these scientists to you, and their ideas to show you how far we've come in these past 100-ish years. Science really, really is amazing. Now, I've already mentioned a couple of these terms, but let's go ahead and formally introduce some terminology. Chemicals that affect body physiology in any manner are called drugs. And chemotherapeutic agents, defined as drugs, act against disease. And antimicrobial agents, or I will just refer to them, and as we've been referring to, antimicrobials, are drugs used to treat infection. And so this is the subject of the rest of this lecture. As we move into the introduction of specific antimicrobial targets for treatment and the control of bacterial infections, I want to formally define for you 2 terms, bacteriostatic and bactericidal. Antimicrobial treatments that inhibit the growth, not kill, but inhibit the growth of a bacteria without causing cell death those treatments are called bacteriostatic. In contrast, antimicrobial treatments that kill bacteria are called bactericidal. Killing by some bactericidal drugs is concentration dependent, where others is time dependent. So we might have a certain concentration of an antimicrobial that leads to death, or we might have a time by which something is exposed, whereby bacteria are killed. Whether an antibiotic simply inhibits growth or kills bacteria often depends on its concentration. The lowest concentration of a drug that inhibits bacterial growth is called the minimum inhibitory concentration. I didn't include it on this slide, but just recognize minimum inhibitory concentration. With a concentration above this, to kill a bacterium called the minimum bactericidal concentration. And generally, the minimum bactericidal concentration is somewhere between like 2 and 8 times the minimum inhibitory concentration. So we have a concentration that inhibits, as well as then a concentration of an antibiotic that leads to death. Finally, on this slide, when we talk about killing a bacteria, killing really isn't defined as wiping out an entire population, but is defined as about a 99.9% reduction of the number of bacteria in a given source or an inoculum. Before we dig into the various ways by which antimicrobials can control bacteria and move on to the next slide, I want to mention the key to choosing a successful chemotherapy agent or antimicrobial against a microbe is first in identifying the microbe itself, and then in relying on a process that we call selective toxicity. That is, an effective antimicrobial agent is selected that needs to be more toxic to a pathogen than to the pathogen's host. And selective toxicity is possible because of differences in structure or metabolism between the pathogen and its host. Typically, the more differences there are between pathogen and host, the easier it is to develop or create an effective antimicrobial agent. And there are many differences between structure and metabolism of a pathogen or a pathogenic bacteria and some kind of a eukaryotic host. And I'll use humans in the examples moving forward. Antimicrobial or antibacterial drugs, antibiotics, constitute the greatest number of diversity in antimicrobial agents. So with that in mind, I want to move to the next slide where we can start talking about the various ways by which we can target a given antimicrobial to a particular process or a particular structure within a bacteria in order to be bactericidal or bacteriostatic. And the first one, antibiotics can interfere with or inhibit bacterial wall synthesis. We know that bacterial cell walls have peptidoglycan. So the major structural component of bacterial cell walls is peptidoglycan. To enlarge or divide, a bacterial cell must synthesize more peptidoglycan by adding those NAG and NAM subunits to existing NAG and NAM subunit or maybe the chains. Many common antibacterial agents act by preventing cross-linkage of the NAM subunits, so interfering with those tetrapeptides. With the most prominent among these drugs, we want to look at the beta-lactams, penicillins and cephalosporins. Beta-lactams inhibit peptidoglycan formation by binding to the enzymes that aid in that cross-linking process of the NAM subunits. And in the absence of correctly formed peptidoglycan layers, the growing bacterial cells have a weakened cell wall with less resistance to the effects of osmotic pressure, water moving into a cell naturally, that cell membrane not having an outer wall with which to bump up against, but instead a cell wall continues to bulge out and bulge out and bulge out, and we see these weakened portions of the cell membrane up against this cell wall, or I should say the weakened portions of the cell wall being pushed upon by the cell membrane as water moves into the cell, and ultimately that leads to cell lysis. So this is one way antibiotics can interfere with or we can focus on control of growth. Now, it's worth mentioning here that inhibiting bacterial cell wall synthesis can work well and fine for bacteria that have a cell wall, but there are a few bacteria, such as Chlamydia and Rickettsia, that lack the traditional cell wall structure we might find in more common bacteria. And because of this, those particular species are more resistant to antibiotics that inhibit bacterial cell wall synthesis. Next up, we have antibiotics that can interfere with cell membranes. Some antimicrobial or antibacterial drugs disrupt cell membranes of target cells, often by forming a channel through the membrane, damaging cell membrane integrity, or by disrupting a transport process across the cell membrane, or otherwise inhibiting the synthesis of some kind of component that's necessary for the cell membrane to remain intact. We have antibiotics that can interfere with or inhibit bacterial DNA synthesis. By interfering with DNA synthesis, drugs such as the fluoroquinolones used to treat some gram-negative bacteria inhibit DNA synthesis machinery, such as DNA gyrase and the topoisomerase enzymes. So by inhibiting the function of some of the machinery needed for DNA synthesis, we prevent DNA synthesis, we prevent the multiplication of bacteria through binary fission. We also have antibiotics that can interfere with or inhibit bacterial protein synthesis. Once inside bacteria, some antibiotics can bind irreversibly to sites on the ribosome or otherwise inhibit ribosomes from undergoing protein synthesis. And in doing so, they can target differences between prokaryotic and eukaryotic ribosomes. Remember, prokaryotic ribosomes have a 70S structure. Eukaryotes have an 80S structure, and our prokaryotes, our 70S structure is made of two subunits, a 30S and a 50S subunit. Because of these differences, antimicrobials that interfere with protein synthesis in prokaryotes like bacteria can carefully and very craftfully, we don't see those antimicrobials causing harm to the ribosomes in our eukaryotic cells. Antimicrobials such as streptomycin and gentamicin can modify the shape of our 30S subunit ribosome in our prokaryotes, making it impossible for the ribosome to read our mRNA correctly, so reading the transcript correctly. Another antimicrobial, the tetracyclines, block part of the 30S subunit such that additional amino acids can't be added when we have those transfer RNAs coming in, and we're going to see how those processes work in a couple of chapters. Some antimicrobials are going to find other ways to target the 50S subunit, and so we'll look at some of those when we're considering protein synthesis. Antibiotics can interfere with or inhibit mycolic acid synthesis, and so this is very species specific. Those species of bacteria that have a heavy mycolic acid cell wall, Mycobacterium tuberculosis is one of those. So the species, unlike other more conventional bacteria, have a cell wall that contains large quantities of lipids. And so some antibiotics can come and inhibit the synthesis of mycolic acid and achieve bactericidal activity against bacteria such as Mycobacterium tuberculosis. Antibiotics can inhibit folic acid synthesis. Again, this is now we're targeting a process or a pathway within a cell, a bacterial cell. So folic acid, is involved in a number of bacterial cell reactions, specifically biochemical pathways leading to the synthesis of proteins and nucleic acids. Ultimately, folate or folic acid is essential for cell survival. So if we rely on an antimicrobial that's called an antifolate, if we use those to focus on targeting enzymes and biochemical pathways, we may inhibit one of those pathways, which then inhibits the production of proteins or nucleic acids. There are a variety of antimicrobials that target these enzymes, and a common one is the sulfonamides. Our last slide of this lecture addresses the clinical conditions and possibility of adverse side effects when prescribing an antimicrobial drug. Ultimately, as a practitioner, we need to consider three things. We need to consider toxicity, we need to consider allergies, and we need to consider the disruption of normal microbiota. Toxicity is our first, and although antimicrobial drugs are ideally selectively toxic against microbes and harmless to hosts, including humans, many antimicrobials do have some toxic side effects, maybe at a certain concentration. Some drugs may be toxic to the kidneys, to the liver, to nerves. Sometimes doctors need to be very careful when prescribing drugs for pregnant women, as drugs are safe. Maybe there are drug studies that show safety for adults, but can't be absorbed or shouldn't be absorbed by the fetus. Maybe there are some drugs that aren't safe for children that are safe for adults. So toxicity is something to consider as a side effect. We need to consider allergies. In addition to toxicity, some drugs trigger allergic immune responses in sensitive patients. And although it's rare, such reactions, these allergic reactions, can certainly be life-threatening, especially in an immediate violent reaction we call anaphylactic shock. Now, we also have disruption of normal microbiota in the body. And so what is our normal microbiota? And the normal microbiota includes all of the bacteria that live and reside in our body in this mutualistic relationship where under normal, healthy conditions, those bacteria live in a way that aren't causing us harm. Drugs that disrupt the normal microbiota may ultimately result in secondary infections. For instance, long-term use of broad-spectrum antibiotics can result in an explosive growth rate of Candida albicans, a fungus found in our vaginal canal that is typically kept in check by resident microbiota genus of bacteria called lactobacillus. They compete for glucose or glycogen that's secreted by the cells in our vaginal canal, and glucose is typically consumed by those lactobacilli and leaving very few or very little bit of nutrients for this Candida species. However, with the use of antimicrobials, sometimes lactobacillus is killed off or reduced, leading to more food and more resources available for Candida, and we have this explosive growth of a fungus within our vaginal canal. We might also see another situation by which wiping out the normal microbiota lends to an opportunistic relationship by which Clostridium difficile or C. diff ends up getting a bit out of control within the colon. This disruption, again, is commonly called opportunistic, and both of these are opportunities by which the normal microbiota aren't functioning properly because maybe they've been wiped out, lending to these other bacteria that have been in very low colony counts growing in our body to explode and take off. Under conditions by which normal microbiota is disrupted, these bacteria or this fungus can grow explosively. So ultimately, this ends our section for module number 3. This concludes the material we're covering at the moment for chapter 20. This goes along with chapter 5 material and control of microbial growth. If you have any questions, please don't hesitate to reach out, and have a great day.